Classification of prokaryotes. Prokaryotes. Prokaryotes are single-celled organisms that have no internal membranes. The easiest identifying characteristics of prokaryotes are 1. They have a single, circular chromosome. 2. They have no defined nucleus or nuclear membranes. Rather, they have a nucleoid, a region of the cell where the chromosome is located. All the diverse organisms that belong to the domains are bacteria and archaea are prokaryotes. Most organisms on Earth are prokaryotes and have dominated ecology over the history of life. Some bacteria and archaea are called extremophiles because they live in extreme environments. Extreme environments are those that plants and animals would find inhospitable. Extreme temperatures, pH, gas composition, or salt concentrations are characteristics of extremophiles ranges. The first living cells were thought to be extremophiles, evolving at high temperature and pressure near hydrothermal vents. Phylogenetic tree of life. In this phylogenetic tree, this black line down here represents the last universal common ancestor that all life evolved from. These were single-celled organisms, like the single-celled bacteria and archaea that are around today. Even though bacteria and archaea are prokaryotes, notice that archaea and eukaryotes are more closely related to each other than they are to bacteria. Bacteria and archaea. While bacteria and archaea share a basic cell structure, they have different cell wall compositions, plasma membrane compositions, ribosomal sequences, and flagella structures. The genes that encode the metabolic pathways of archaea are more closely related to our own, indicating a closer evolutionary relationship. Domain bacteria branched off the evolutionary tree at an earlier time point. Bacteria have more metabolic pathways that are unique to that lineage. With few exceptions, archaea are not present in the human microbiota, and none are currently known to be associated with infectious diseases in humans, animals, plants, or microorganisms. However, many play important roles in the environment and may thus have an indirect impact on human health. Quoted from Lumen Learning. The remainder of this section will focus on bacteria because they are important to human health. For reference, visit the website shown here. Classification of bacteria. Bacteria are all prokaryotes, yet the millions of species are each unique. Bacteria can be classified on the basis of their shape, whether they grow as single cells or in groups, the composition of their outer layers, and their genome sequences. The three basic shapes of bacteria are coccus, or spherical, 5 times 10 to the minus 7th meters in diameter. 2. Bacillus, or rod-shaped, 5 times 10 to the minus 7th to 2 times 10 to the minus 6 meters in length. And 3. Spiral, which are 1.5 times 10 to the minus 6 meters in length. Examples of each shape are found on the following slides. Spherical bacteria. Gonorrhea, a human sexually transmitted infection, is caused by a pathogenic diplococcus, a spherical bacterium that exists as pairs of cells. Picture A, shown here, is a gram stain of N. gonorrhoeae from a pure culture. Picture B shows a gram stain of a pustular exudate of N. gonorrhoeae in neutrophils. And picture C shows a scanning electron micrograph of N. gonorrhoeae diplococci. These photos are courtesy of Chris T. McAllister. The reference link is behind the image. Rod-shaped bacteria. Escherichia coli, more commonly known as E. coli, is an organism that is common in our bodies and in our environment, and is commonly used as an experimental model. We have learned a lot about bacteria by studying this rod-shaped species. It is typically not dangerous unless we ingest large doses of it, which results in food poisoning. Spiral bacteria. Syphilis, another sexually transmitted infection, is caused by a spiral bacteria, Treponema pallidum. Classification of bacteria. Another way to classify bacteria is by the thickness and composition of their peptidoglycan cell wall. As you will learn in the next section, The outer layers of bacteria are a lipid membrane, surrounded by a peptidoglycan cell wall, and sometimes surrounded by a capsule. Gram-positive bacteria can be stained with a gram stain that sticks to their peptidoglycan, while gram-negative bacteria have thin walls and do not retain the stain. The image to the right shows an enlarged photograph of gram-positive cocci and gram-negative bacilli. The gram stain is blue. Why is the thickness of the peptidoglycan cell wall useful information? The antibiotics we use to kill bacterial infections are different for gram-positive and gram-negative bacteria. These types of bacteria also differ in how easily they dry out, how easily they are killed with certain detergents, and whether lysozyme, an enzyme in tears, blood, and some white blood cells, can kill them. The simple gram stain test helps medical professionals select the right treatment for a pathogenic bacterial infection. The ultimate tool for classifying bacteria is DNA sequencing. Microbiologists sequence a particular gene, 16S rRNA, to identify different species of bacteria in complex mixtures of cells. Each species has a unique 16S rRNA gene sequence, which can be aligned with other 16S rRNA sequences for comparison. The more similar the 16S rRNA gene sequences, the more closely related are the species.